Okay, time jump. For you, you're just starting the next video. For me, it has been a long day. But whatever that is, whatever. So we just finished talking about the lytic cycle. The lysogenic cycle is a little bit different. This is the cycle, remember, where the viral chromosome gets incorporated into the bacterial DNA. And that's what you're looking at here. Infection occurs, and then instead of taking the choice of destroying the cell and making new minions of its own, the chromosome somehow gets the signal to incorporate into the bacterial chromosome, and where it becomes a prophage. So a prophage is basically viral DNA living in a living cell's DNA. As the cell replicates and undergoes binary fission, the cell divides in half, right? And then that cell divides in half. Remember, this is not meiosis. This is binary fission. They're just showing 4 cells. And the cell will, as the cell replicates every time, it makes new DNA, makes new viral DNA with its own DNA. Occasionally, the prophage will excise itself out of the bacterial chromosome, and the cell can then enter the lytic cycle. So you'll see a situation here where this is like what happens with human papillomavirus, with HPV. There are a number of different viruses, herpes simplex virus, the herpes simplex virus and varicella virus. You'll see that the prophage is integrated into the human's DNA. It's a little bit different because that's a human disease, and it'll just live and live and live in you until something happens, usually a stressful event, and the viral DNA excises itself out, and then it hits the lytic cycle. So good times, you know, especially if you're the, well, not good times if you're the one that has it. So what happens in eukaryotes? That's a little bit different. Well, we do have, again, viruses that can become prophages. Another type of virus in eukaryotes are retroviruses, RNA viruses that make DNA, which then go through the whole lytic cycle. And so one of the things that's really important in the viral genome that in retroviruses is an enzyme, the sequence or the gene for an enzyme called reverse transcriptase. Reverse transcriptase takes DNA and converts it back to takes RNA and converts it back to DNA, and that allows the cell to go on and not really know that it's sick for a little while, because the RNA has to be made, viral RNA needs to be made into viral DNA, and then the infection can occur. You can also get a situation where a provirus, which is like a prophage, but a provirus integrates in the host chromosome. And this can sit in the cell for years before it gets pulled out. So, I mean, that's kind of a crazy thing, right? And so here's an example of viral RNA inside of a eukaryotic virus. In this case, it's the HIV virus. And what we're looking at here is the HIV virus, the spike glycoproteins, identifying receptors on the T cell. The outside envelope, the viral envelope, merges with the cell membrane, releasing the virus itself. The viral coat, the capsid breaks down, releasing the RNA with this little bit of basically the enzyme that's going to make the RNA to DNA. I'm sorry, it's been a long day, guys. And then the RNA makes DNA. DNA then goes into the nucleus, where it can either become a provirus using an enzyme called integrase, or sometimes this will stay out in the cytoplasm, allowing killing the cell, basically replicating the virus. Or what's even worse, sometimes the RNA will stay out here and the virus will stay inside the cell, and the cell will shed virus. Virus will actually leave the cell. That happens a lot of times with like flu virus and rhinovirus. You can shed virus for a period of time. So the reproductive cycle, This is really important. It's important to understand the reproductive cycle, because if you can interrupt the reproductive cycle, then you can actually interfere with the virus replicating it and making the person sick. And so we have the transcription of viral genes. Okay, so viral genes being made into RNA and then protein. That's the transcription, making RNA. That was actually on your exam, remember that? The first step in expressing a protein is transcribing RNA and then translating it into a protein. That is, obviously protein. So basically, retrovirus itself has to go from RNA to DNA, then back to RNA for the cycle to complete, or it becomes a provirus, and that piece of DNA, instead of making new RNA, gets incorporated into the host genome. DNA viruses are pretty simple. Actually, they're real simple, similar. Some of the simple ones are similar to a phage, where DNA viruses undergo DNA replication, RNA is made, and then you have destruction of the cell. So, when the cell is going into the lytic cycle, even in human viruses, you have a situation where the virus has to assemble in the cytoplasm of the cell. So we've talked about infection. We've talked about, what's going to happen. Is it an RNA making DNA, or is it DNA going in there? And is it becoming incorporated in the chromosome? Let's now say we're going lytic here. Protein and nucleic acids come together to make the infectious particle. This is your virus, right? Oops. Some viruses are able to self-assemble. Some viruses are just really good at making themselves, copies of themselves. Some viruses use proteins to help them assemble. In other words, they have proteins that kind of work as a factory to make more virus. And these aren't normally found in cells. So the reproductive cycle involves release, and then lysis. So back to bacteriophage. The viral cell cycle ends with that release/lysis. In other words, we say release in human cells. This is also shedding. So the cell might be alive, manufacturing more viral particles, but the viruses are shedding off the cell. Remember when we were back here looking at this picture, we saw this outer membrane that fuses with the cell membrane right here. This process of going in can also go backwards, and you'll have this portion of the virus taking a piece of the cell membrane with it. And that's what the shedding event is. It's kind of, it's kind of gross, but it's kind of interesting at the same time. Eukaryotic cells bud, or the viruses bud or shed. Budding and shedding is the same thing. Okay, from the cell. And this is where the envelope of the virus comes from. It's actually a chunk of the cell membrane from the previous cell that had been infected. And that cell membrane actually has new proteins on it that allow that virus to have new traits. It's kind of a way for the virus to learn new things. So one of the first viruses that was studied was bacteriophage T4. T4 is still used for lots of research for a variety of manipulations and identifications of other genes and stuff. Usually it infects E. coli, so since the E. coli is a very common research tool, T4 is also a common research tool. It has quite a few genes, 300. I know we say a large number of genes, but for a virus, that's a lot of genes. And it allows us to study genetics because it allows you to see how genes move around from cell to cell via infection. It's basically the E. coli T4 system is a virulent phage that is a model system for studying genetics. It has no latent or lysogenic stage. And so here's an example. Here's the genetic material. Here's the protein head. This tunnel here is the shaft. And then you have the tail fiber. And then the base plate here is what contacts or gets into contact with the cell, where the virus holds onto the cell. E. coli cells grow really well on an agar plate in Petri dishes. And what's really convenient about E. coli cells is that they grow well, they grow quickly overnight. A good thing is that they can grow into what we call a lawn. You can spread E. coli, you can actually start with just a small amount of E. coli, spread it over the plate, and by morning, the whole plate will be covered with bacteria. So that's the upside. You have lots of individuals. The downside is you can't distinguish one cell from another. In other words, they all look the same. So as long as you think you have a colony that, or at least a group of cells that came from the same individual, you can assume that maybe they were all clones. And so when the cells are growing, they'll end up touching each other, and it gets kind of crowded on the plate, so it looks hazy or opaque. Now, when they're growing on the agar, you have a nice kind of milky looking, it's kind of yellowish looking surface. When you add a virus to it, cells will become infected. All right, I probably have a picture here. Yeah, I'll wait till the picture. I can draw a little bit. Your cells will become infected, and little holes will develop where viruses infected cells, because remember, the first cell is going to burst, and that is going to release phage, and that's going to infect neighboring cells. And as they burst, go through the lytic cycle, they'll release phage. And so basically you have a wave of death proceeding through the plate. And so as long as you have cells that are not infected surrounding cells that were infected, you have the potential for spreading the infection. So the lysed area looks clear compared to the unlysed area. And so here's an example of that. Here's a bacterial Petri dish. This is kind of what it looks like. It looks kind of hazy. If you looked at the bacterial Petri dish using a microscope, you could see that you have lots and lots of little bacteria there. Here's some phage DNA, a phage capsid. Here, it's a drawing, but and you can see where the initial infection takes place. That first cell is infected. It undergoes the whole lytic cycle. Release occurs, rupture, lysis, pushes all of these little phages out into the environment, where they infect the next line of cells and the next line of cells. So what ends up happening is you'll have what they call plaques. Plaques are the clear areas, and the cloudy areas are lawns. So each of these little yellow things aren't actually mounds of bacteria. They're actually areas lacking bacteria. They're circles with no bacteria in them. And so changes in the bacteriophage can cause a change in what a plaque looks like, and we call that plaque morphology. So there are different types of T4 phages. The wild type makes these like little tiny, plaques. They're kind of small. Plaques caused by rapid lysis. So in other words, there are plaques that work rapid lysis phage. Those bacteriophage strains kill cells even faster than the normal wild types do, and so you get more death in the same amount of time. So you see the zone of infection spreading farther. And so to test for mutations in plaque morphology, what you do is basically watch what the plaques look like.
Natalie needs you.
Sorry about that. I was needed real quick in the other room. Okay, so let's say, for example, you have two mutant strains infecting a single cell. One type of no wild type plaques. If you don't have If you don't get wild type plaques from that particular infection, you have something called non-complementation. And what that means is that the mutations are in the same gene. Non-complementation just simply means that if you have a mutant in a single gene that gives rise to a shape of a plaque, and it didn't change the shape of the plaque, the same gene was affected. If the wild type plaques, however, have a different shape, then that's complementation. And that means that each strain is different, or each strain has a mutation in different genes. So non-complementation, mutation in the same gene. Complementation, mutation in different genes. It's actually pretty easy. So here's an example of an experiment where the RLL strain 1, here's gene A and gene B, the E. coli, and RLL strain 2. You basically have two genes. You didn't, the plate observed, you want to observe if plaques were formed. No plaques were formed, and that means that no complementation occurred because you didn't have mutation in the gene. So this experiment, and here's complementation, you end up with viral plaques because you have two separate genes. Oop, wrong pen. Two separate genes that are affected. That's all you really need to know about that. So we'll pick up on mapping in just a few minutes.